Developmental biology of the embryo. Key milestones in early development. The development of an embryo involves a series of highly coordinated events that transform a fertilized egg, zygote, into a complex multicellular organism. These early milestones are critical in setting the foundation for the formation of tissues, organs, and the overall body structure. From fertilization to the formation of the basic body plan, each milestone plays a pivotal role in shaping the developing embryo. 1. Fertilization and zygote formation. Fertilization marks the union of the male sperm and the female oocyte egg, resulting in the formation of a zygote. The sperm and egg nuclei fuse, restoring the diploid chromosome number, 46 chromosomes in humans. Cortical reaction. Upon sperm entry, the egg undergoes a cortical reaction to block additional sperm from fertilizing the egg. Zygote formation. The fertilized egg, now called the zygote, contains genetic material from both parents, which will direct the development of the embryo. 2. Cleavage. Early cell division. Cleavage refers to the rapid mitotic cell divisions of the zygote that result in the formation of smaller cells called blastomeres. The first cleavage occurs about 24 hours after fertilization. Cleavage divisions occur without growth, so the overall size of the embryo remains the same as it divides. Cleavage is holoblastic in humans, meaning the entire egg divides into smaller cells. After several rounds of division, the embryo forms a morula, a solid ball of cells, and later a blastocyst. Third, formation of the morula and blastocyst. Morula, the solid ball of cells, about 16 to 32 cells, formed by cleavage. This stage occurs around 3 to 4 days after fertilization. The morula moves from the fallopian tube towards the uterus. Blastocyst formation. By day 5 or 6, the morula develops into a blastocyst. This process involves the formation of a blastocoel, a fluid-filled cavity, and the differentiation of cells into trophoblast, the outer layer of cells that will contribute to the formation of the placenta. Inner cell mass, ICM, a cluster of cells inside the blastocyst that will develop into the embryo. Viator implantation. Implantation occurs around 6 to 7 days after fertilization, when the blastocyst attaches to the endometrial lining of the uterus. The trophoblast cells invade the endometrial lining, and enzymes are secreted to facilitate embedding. The syncytial trophoblast layer formed by trophoblast cells is responsible for placenta formation and nutrient exchange between mother and embryo. Successful implantation marks the beginning of pregnancy and establishes the early placental connection for nourishment and waste removal. Fifth, formation of the blaminar germ disc. After implantation, the inner cell mass of the blastocyst differentiates into 2 distinct layers. Epiblast, the upper layer that gives rise to most of the tissues and structures of the embryo. Hypoblast, the lower layer that contributes to extraembryonic tissues such as the yolk sac. These layers form the bilaminar germ disc, a precursor to the 3 primary germ layers. 6. Gastrulation, formation of the 3 germ layers. Gastrulation occurs around day 14 to 16 after fertilization and is a critical process in early development. During gastrulation, the bilaminar germ disc forms the 3 primary germ layers. Ectoderm forms the skin, nervous system, brain, spinal cord, and other structures like hair and nails. Mesoderm forms muscles, bones, heart, blood vessels, kidneys, and reproductive organs. Endoderm forms internal organs such as the lungs, liver, pancreas, and gastrointestinal tract. Gastrulation results in the formation of the trilaminar germ disc, marking the establishment of the body's basic structural plan. 7. Neurulation, formation of the neural tube. Neurulation begins during the 3rd week of development following gastrulation. Neural tube formation. The ectoderm above the notochord thickens to form the neural plate, which folds to form the neural tube. The neural tube eventually develops into the central nervous system, brain, and spinal cord. Neurulation is a key milestone in the development of the nervous system, establishing the foundation for further development of the brain and spinal cord. 8. Organogenesis, formation of organs. Organogenesis refers to the formation of organs from the 3 germ layers, which begins shortly after gastrulation and neurulation. The ectoderm forms the brain, spinal cord, skin, and sensory organs. The mesoderm forms muscles, bones, kidneys, heart, and blood vessels. The endoderm forms the lungs, liver, pancreas, digestive tract, and other internal organs. This stage marks the beginning of the formation of the body's organs and systems, and it is crucial for the development of a functional organism. 9. Formation of the extra-embryonic membranes. During early embryonic development, the extra-embryonic membranes are formed, including amnion surrounds the embryo, forming the amniotic sac that cushions and protects the developing embryo. Chorion contributes to the formation of the placenta and facilitates nutrient and gas exchange. Yolk sac, involved in early blood cell production and later contributes to the digestive system. Allantois forms part of the umbilical cord and contributes to waste removal. These membranes are essential for protecting and sustaining the embryo during development. 10th conclusion. Early milestones of embryogenesis. Embryogenesis progresses through a series of key milestones: fertilization and zygote formation. The union of sperm and egg forms the zygote. Cleavage. Rapid cell division produces a morula. Blastulation. Formation of the blastocyst. Implantation. The blastocyst embeds into the uterine wall. Gastrulation. Formation of the 3 germ layers. Neurulation. Formation of the neural tube. Organogenesis. Formation of organs and tissues. These milestones are critical for the proper development of the embryo and set the stage for further fetal development.